Uh, Let us start the chapter 4th, Plant Genetic Engineering. Genetic engineering. It means that whatever the genetic manipulations or any change in the chromosomes or alteration of the chromosomes or alteration of the genes or the genetic manipulation, simply it is called as genetic engineering. The concept and the definition. The genetic engineering is defined as the direct and artificial alteration of genetic makeup of cell or organism, either through modification of specific gene or by implantation of the new gene into the chromosomes. It means that the genetic scenario is changed by the help of the genetic engineering. Whatever the number of the chromosomes, number of the that genes which is responsible for the particular character, that can be directly changed. And this change that is done or it is made by the help of the either manipulation or by modification of the particular type of the gene or a particular type of the chromosome within the that organism. It is the technique of transfer of gene from the one organism to the another. At present, it is also known as the recombinant DNA technology or the gene cloning. The genetic engineering involves the manipulation of DNA to get the desired goal of predetermined way. What we exactly want? We can directly change the total makeup of the that organism. That is the desired character, this desired character that we should bring it in the that new organism, or the new organism that will be directly changed or formed by the help of the manipulations of the particular type of the gene. The gene is responsible for the particular type of the characters, and these manipulations, either by manipulation or by alteration or by modification of the particular type of the gene within the organism, it is simply called as a genetic engineering. The Cohen and the Boyer were the first to cut the DNA into the region of the fragments, into the E. coli bacteria. The basic principle of the recombinant technology is the manipulation and alteration of the gene. The basic principle regarding this recombinant DNA technology, it is nothing but the manipulation and the alteration of the gene. But this manipulation and the alteration of the gene, it is carried out by the help of the alteration or by modification. The tools of genetic engineering. There are the three basic tools of genetic engineering or DNA technology. That is, first, restriction enzymes, second, linkage, and third, that is the plasmid vector. The restriction enzyme that we will start first. Restriction enzyme that is called as the endonuclease enzymes. These restriction enzymes that can directly detect the site of the that nucleotide sequence, and that can directly take the cut to the particular site where it has been taken the position. It means that the bacterial enzyme which cut the DNA at the specific nucleotide sequence is known as the restriction enzymes or the restriction endonuclease. They are used for making rDNA sequence analysis of DNA and cloning. The amplification of the gene, there are three types of the restriction enzymes such as class 1, class 2 and the class 3. The class 1 and the class 3 are very similar, but class 2 is a different class. The class 2 is the restriction enzyme that detects the specific site of the nucleotide sequence and cleave the DNA at that specific site. It means that the class 2 restriction enzyme that can directly detect where the cut should be taken on the particular type of nucleotide sequence. And then it can directly cut that nucleotide sequence where the position were taken by itself. It can directly cut that nucleotide sequence where the site were introduced or site were directly detected by itself, and the cleavage that it starts. After cutting the product, it produces the end with the single strands of DNA overhangs known as the sticky ends, which helps in the cloning as they can be linked by DNA linkage. Ligase, that is a sort of the enzymes, the ligase, it is also known as the DNA ligase. If two DNA molecules have matching end are joined by the enzyme DNA ligase, which seal the gap between the molecules, forming the single piece of DNA. When these cut pieces of the DNA nucleotide sequence that has been formed, the cut pieces of the DNA, they are directly sealed with the help of the particular type of the enzyme, that enzyme that is called as the ligase enzyme, and this ligase enzyme, that is the DNA ligase enzyme, that can directly seal. It means that it stick the both ends of the that nucleotide sequence of the DNA. For this ligase, use the energy in the form of the ATP and catalyze the reaction in which the phosphate group of the stick ends of one DNA strand linked with the hydroxyl group of end of the tDNA, T4 DNA ligase is commonly used, which first isolated from the E. coli. Second, that is the plasmid vector. This is commonly known as the cloning vehicle. This is known as the cloning vehicle. It means that whenever the gene cloning it has been process that has been started, or whenever the gene cloning process is supposed to be started, at that time it is necessary to have the particular type of the vehicle. The cut pieces of the nucleotide sequence, which is the desired characters. The cut pieces of the nucleotide sequences which are in the desired character. These desired characters of that gene is directly set in the entered that cloning vehicle, that is the plasmid vector, and this cloning vehicle which is directly implant in the enter the E. coli cells, and that can be directly genetic engineering that is supposed to be complete. Now this plasmid vector, it is commonly known as the cloning vehicle. It is circular double-stranded DNA molecule present outside the main chromosomes in the bacterial cell. The plasmid used in the genetic engineering is called as the vector for the transfer gene from one organism to the another. The main feature of the plasmid, that is characters the main feature of the plasmids, first, most of the plasmids are circular double-stranded DNA molecule. Second, they replicate independently in the cell. They can replicate. They are having the ability to replicate. When they will replicate, it will give directly the gene library. Replication, that it means the multiplications of the particular type of the nucleotide sequence, and these multiplications of the particular type of nucleotide sequence that will form the pieces, that is the fragments of the DNA, desired DNA, and these fragments, that is the gene library. And third, they depend upon the host enzymes for the replication. And fourth, they are capable of introduce the foreign gene into the cell, which then acquire the new characters. Foreign DNA. If foreign DNA, that is our desired DNA, this foreign DNA is supposed to be implanted in the entire that host DNA. At that time, the cloning vehicle is essential, and this cloning vehicle, that is in the form of the plasmid vector. They produce the multiple cloning sites. PBR322, PUC vectors, East plasmid vectors. These are the common type of the entire that plasmid vector. TI plasmid are some of the important plasmid vector. PBR322 is the first artificial vector constructed and used for the cloning. These plasmid contain 20 unique recognition sites for the restriction enzyme. TI plasmids are larger circular DNA molecules. They exist. TI plasmid means tumor-inducing plasmid. They exist in the bacterial cell. Most of the cloning vehicles in the plant are based on the TI plasmid. It belongs to the soil bacterium, that is the Agrobacterium tumefaciens. This Agrobacterium tumefaciens is the soil-borne bacterium, and this tumor-inducing plasmid, which is present within this Agrobacterium tumefaciens, a type of bacteria that has the origin of replication, the conjugations, and the gene responsible for the catabolism of the opians. Gene cloning. How the gene cloning is made? It means that whatever the desired genes that has been obtained by the help of the restriction enzymes, that restriction enzymes will make the cut where the particular site of the nucleotide sequence, and whatever the entire the fragment that has been formed by the help of the restriction endonuclease, whatever the fragment that has been formed by the help of the restriction enzyme, These DNA, that is the foreign DNA, that can be directly attached to the entire that host cells along with the plasmid vector, along with the cloning vehicle. At that time, the gene cloning is supposed to be start. Now, the production of the exact copies of the DNA sequence using the genetic engineering technique is called as the gene cloning or the DNA cloning. The gene cloning is the process of the isolating of DNA sequence of interest for the purpose of making multiple copies of it. The basic type of the gene cloning is in vivo gene cloning. In this type, the restriction enzymes, ligase, plasmid vector fragments of the DNA are made by the help of the cloning in the host cells. It is also called as cell-based cloning. Second, in vitro gene cloning. It involves the polymerase chain reaction, that is the PCR technique. The polymerase chain reaction methods to copies the fragmented DNA without vector and the host cells create the copies. Steps in the gene cloning. First steps, isolation of the DNA, that is desired DNA, that is gene of interest, fragment into the cloned, to be cloned. It means that isolation of the DNA. Whenever which type of the gene which is responsible for the particular character, if that gene is isolated and that isolated DNA of the desired traits, for example, one of the wheat variety is there Kalyan Sona and second wheat variety is there the 2189. If the particular characters of the Kalyan Sona, that is the yellowness of the characters of the Kalyan Sona, if it is supposed to be bring in the 2189, it is possible by the help of the genetic engineering. Now the character, that is the yellowness of the Kalyan Sona, that is the character which is responsible, which is restricted to the particular gene. It means that the particular gene is responsible for a particular character. So the gene responsible for the yellowness of the Kalyan Sona, this can be directly isolated from the Kalyan Sona by the help of the restriction enzyme, restriction endonuclease enzyme, and it is directly implanted in the entire the 2189, ज्याला आम्ही 2189 गहू म्हणतो, त्या गव्हामध्ये जर हा जीन्स टाकायचा जर झाला, if it is interested, if we are interested to bring that yellowness of the Kalyan Sona into the 2189, it is possible by the help of the genetic engineering. For that purpose, we will have to isolate that the gene which is responsible for the yellowness of the Kalyan Sona from the Kalyan Sona plant, Kalyan Sona wheat plant, or the Kalyan Sona wheat cells of that plant, and then it is to be isolated, and then the isolated DNA, it is to be purified by the help of the gel electrophoresis and whatever it may be. Second, that is the insertion of the isolated DNA into the vector to form the recombinant DNA. Now this isolated DNA, that is our desired traits, what is our desired traits? Yellowness of the Kalyan Sona. Yellowness of the Kalyan Sona, it is our desired traits, and gene responsible for that, take out that genes, and that insertion of that DNA which is responsible for the Kalyan Sona into the vector, that is the plasmid vector. It may be the PBR22, it may be the what we used to say, that is the yeast plasmid vectors or the TI plasmid, whatever it may be. Now, this insertion of the isolated DNA into the vector by the different from the recombinant DNA. And third step, that is the introduction of rDNA into the host cells. Now, the introduction to introduce this rDNA, that is the recombinant DNA. What is the recombinant DNA? That recombinant DNA, that is nothing but the desired genes. The genes responsible for the particular type of the entered character. Now take that particular gene responsible for the particular type of the characters along with that DNA. It means that DNA sequences that is responsible for the yellowness. Now that recombinant DNA, it is directly introduced in the host cell. What is our host cell? 2189 cells of the 2189, it is our host cells. And the fourth step, selection of the transformed host cells and identification of the clone containing the desired genes. Now fifth, that is the amplification, that is the multiplication. Now the amplification or the multiplication of the recombinant DNA, it is a very important stage, that is the enter the genetic engineering as well as that is in the gene cloning. For that purpose, if the more amplification, if there is a more multiplication of the recombinant DNA, at that time the total character that will be very visible. That visible characters, it means that yellowness character that will be visible if if the such characters which is having the DNA, this DNA which will be multiply itself within the host cell, that is nothing but the amplification. And lastly, isolation of the amplified gene copies by the gene and purification of the isolated gene by the help of this particular method. That is the gel electrophoresis, agarose gel electrophoresis apparatus. This directly gives rise to the total the purification of that DNA or the purification of the isolated gene. Now the application of the gene cloning. The first application, that is the particular gene can be isolated and its nucleotide sequence are determined. Second, mutation can be identified. If there is a mutation within the chromosomal mutations or the gene mutation, what type of the mutations are there? What type of the mutation which is caused by the help of what? By whom the mutation has been formed? Who are the responsible for the particular type of the mutation? Whether the chromosome is responsible at that time, that chromosomal mutation that has been formed, or the gene mutation. The mutation can be identified by the help of the gene cloning. And third one, that is the organism can be engineered for the specific purpose. What is the specific purpose? What is our purpose? Our purpose is that the yellowness of the enter that Kalyansona should be bring in the enter that 2189. This can be done by the help of the enter that gene cloning, by the help of the genetic engineering. PCR, that is the polymerase chain reaction. It is the polymerase chain reaction invented by the Mullins in 1983. It is a rapid method of the gene cloning. Important steps in the PCRs are: first, primary design; second, the performing PCR; and third one, that is the sequencing PCR; and fourth one, that is the analysis of sequencing data; fifth, assessment of the gene expression. The real-time PCR, comparative PCR, RT-PCR, PCR in situ are the different type of the polymerase chain reaction. Advantages of the PCR technique. What are the advantages of PCR technique? पहिला फायदा त्याचा असा आहे, the first advantages is that this cloning technique is very fast and efficient. Nowadays we are very familiar with the COVID-19. Now within the enter that COVID-19, if the patient is suffered by the help of the COVID-19, at that time the test is taken, that is the RT-PCR technique. And this RT-PCR technique by which the gene cloning responds very fast. With this very cloning technique, it is very fast and efficient. We can directly confirm that whether the patient is suffered by the corona virus by the help of the PCR technique, RT-PCR technique. It is formed or it is done by the help of this, and it is very fast method. It is done by the help of the PCR technique. Second, that is the highly sensitive with the potential to produce the millions of the copies of the specific product. Millions of the copies of the specific product that has been formed by the help of the polymerase chain reaction technique. It means that whatever they enter that copies of the enter the gene sequences, copied gene sequences are formed by the help of the enter the RT-PCR technique, and this is the best and very advanced advantages of the enter the PCR technique concern. It is very simple to use and gives result very rapidly. आपल्याला लगेच त्याचा रिझल्ट मिळतो. Second, that is it is useful in the amplification and the quantification of the DNA. And fifth one, that is the PCR based DNA fingerprinting has a wide application in the forensic laboratory, where there is wide scope in the enter the RT-PCR technique or the PCR technique, polymerase chain reaction technique, which has a wide scope in the forensic laboratory. PCR have the huge medicinals and the diagnostic applications in the bacterial as well as the viral diseases. Just now I have been told you regarding the COVID-19. This technique, which has a very medicinal and the diagnostic application, it can be diagnosed whether the patient is suffered by the help of the coronavirus or not. By the help of the entered polymerase chain reaction technique, it is possible which type of the entered virus or bacteria is there. That can be directly identified by the help of this PCR technique concern. Disadvantages सुद्धा आहे त्याच्या असं म्हटलं तर some disadvantages are also there. It is having the limited vector choice. For these, vectors that is very limited. A particular vector is responsible for the RT-PCR technique, or the particular vector is also a type specimen of the entire particular vector that is also responsible. Then it is higher cost, directional cloning is difficult, lack of sequence control at junction, and the multi-fragment cloning is not straightforward. Application of the plant genetic engineering. Plant genetic engineering has a great importance in the industrial as well as the agricultural sector. It has enabled the genetic manipulations of the different economically important plant. Genetically modified organisms or the transgenics are the new hopes of the 21st century. The development of the transgenic plants in the result is the integrated applications of the rDNA technology, gene transfer methods, tissue culture technique which are used in the entire the development of new economically important traits. For example, insect pest resistance, delayed fruit ripening, abiotic stress, etc. are the some application of the genetic engineering.